Hello, everyone. We're Group 11, and this is our presentation of our intra-blood-brain barrier glutamate monitor and anti-epileptic drug delivery system. Epilepsy is a chronic neurologic disorder characterized by recurrent seizures and the associated cognitive consequences. According to the WHO, over 50 million people suffer from various forms of epilepsy worldwide. But despite decades of clinical research, there's still no cure, and about a third of patients don't respond to typical therapies. This is in part due to the very etiology of the disease, but essentially there's a poor understanding of the exact electrochemical dysfunctions that lead to the hyperactivity of neurons. To understand the pathophysiology of epilepsy and seizures, it's important to examine the part of the brain that is affected. Seizures are abnormal, excessive, hypersynchronous discharges of a population of cortical neurons and can be focal or generalized. The cerebral cortex is the outermost portion of the brain that controls many of our higher order sensory and motor functions, and it's unique because it contains a vast network of excitatory neurons. The primary neurotransmitter for these neurons, and in fact the most abundant neurotransmitter of all, is glutamate. Glutamate is an amino acid that is made in the brain and cannot cross the blood-brain barrier. It can bind with several types of ionotropic receptors that promote action potentials. Our interest in creating a biosensor for glutamate stems from a study that found elevated levels of extracellular glutamate in the brains of patients with spontaneous seizures. The image on the right shows this in an MRI contrast study. A major concern is that excessive levels of glutamate can lead to excitotoxicity, a state in which the postsynaptic neurons are overwhelmed by the excitatory signals and ultimately die. In fact, this is believed to be a major contributing factor in neurodegenerative diseases like ALS and Alzheimer's. In a healthy brain, excitation is balanced by signals directed by inhibitory neurotransmitters like gamma-aminobutyric acid, or GABA, and anti-epileptic drugs work by the same principle.
We are using clonazepam for our device. Clonazepam is a benzodiazepine with marked anti-epileptic properties. Benzodiazepines are a class of drugs that calm or sedate individuals by increasing the level of GABA in the brain. If someone does not have enough GABA, their body may remain in an excited state, causing seizures. Clonazepam is prescribed for daily usage, but we chose it for this project because it is often used in the emergency management of status epilepticus. For emergency treatment, 1 milligram is given via slow intravenous injection over a time span of at least 2 minutes. This is then repeated as necessary for up to the maximum daily dosage of 20 milligrams.
Current treatments for refractory epilepsy are largely centered around nerve stimulation devices. These options function as brain pacemaker-like systems to manipulate electrical pulses in the brain. Limitations of these devices are numerous. Several are very invasive due to size and multiple components, while others can take years in the body to be fully functioning. And all are palliative procedures that rarely stop seizures completely. Due to the link of glutamate to epilepsy, glutamate reading biosensors are also of interest. One in development has shown promising cost and sensitivity, linearity, and selectivity. A limitation of this design is that it is intended for implementation into the spinal cord, meaning it would not read glutamate levels directly in the brain. Also, this device only monitors for glutamate levels while having no function to improve them. Based upon these limitations, there is a need for a device that is small, located in the brain, and stops seizures completely.
Next, we will discuss our device and justifications for our design. Our device should meet the following objectives. The biosensor should be flexible and have small dimensions to prevent unwanted pressure in the brain and aid in better tissue integration. The device must be durable and non-biodegradable so it does not deform or lose performance functionality over time. The sensor must be biocompatible to prevent any adverse effects after implantation, such as inflammation or scarring. And finally, the device should be easy to manufacture and cost-effective so it can be marketed at a reasonable price. Our device will detect the levels of glutamate in epileptic patients to determine the maximum glutamate threshold that leads to seizures. Then it will release clonazepam when the threshold is detected. Our device has two main parts: an intra-blood-brain barrier implantable glutamate sensor and an on-body subcutaneous drug delivery system located on the arm. The point on the diagram with the star is the electronic receiver during the 30-day diagnostic period to determine the glutamate threshold. Once the threshold is determined, the receiver can be removed and the drug delivery system will function as a receiver. The release of clonazepam by the drug delivery system would upregulate GABA production to inhibit a seizure at the programmed patient-specific glutamate concentration level. Our biosensor is an amperometric sensor which monitors current generation when electrons are exchanged between a biological receptor and an electrode. The biosensor has a polymer base with 3 electrodes. The working electrode is where the current is measured. The reference electrode is used to measure the working electrode by maintaining a constant electrochemical potential, and the counter electrode is an inert conductor to complete the circuit. Our device has an applied potential of 0.5 volts with reference to silver chloride to obtain the current reading. Based on dimensions of similar intracranial devices, ours will be 30 millimeters by 5 millimeters with a thickness of 78 micrometers. The sensor will be surgically implanted in the subarachnoid space in the brain through a robotic procedure. The subarachnoid space is optimal because it contains cerebrospinal fluid channels, which contains concentrations of glutamate that our device can detect. The current will be transmitted to the electronic system located behind the ear, which includes a signal amplifier and microprocessor. An app on a phone will be able to read the transmitted data from the electronic system by placing the phone in close range. The app will display the data in a visual format, such as a graph of the continuous glutamate readings over time. After 30 days of continuous readings, a maximum glutamate threshold can be determined based on the correlation of the glutamate levels at the time that the patient had a seizure. The device can be 3D printed using sterilizable biocompatible materials. The base of the biosensor is made of PDMS because it will insulate the device and is biocompatible. The working electrode has a top layer of the enzyme glutamate oxidase to catalyze the reaction with glutamate. The bottom layer is the conductive polymer, abbreviated as PE.PSS, and platinum nanoparticles. Compared to metals, this polymer is more flexible, reducing mechanical strain on the tissue, and can be 3D printed relatively easily. Platinum nanoparticles will be used in this layer because they promote oxidation reactions to produce a change in current that can be detected. The reference electrode is made of silver chloride because these molecules have stable half-life potentials that do not change over time. The counter electrode is made of platinum because it is an inert material that can carry current. The biosensor is coated with a BSA solution to protect and immobilize the glutamate oxidase.
Our drug delivery system will consist of biocompatible lightweight materials and has 3 components. A high-density polyethylene base chosen due to its high impact that will last for 30 days at a time, a cyclic olefin copolymer drug pod chosen due to its shatter and chemical resistance and used in similar on-body injectors that will last until the drug is gone, and 3, an ultra-high molecular weight polyethylene receiver chosen due to its abrasion, corrosion, and shatter resistance and used in long-term medical equipment, which will last for 1 year with the biosensor implant. Other important materials will include a waterproof, safe adhesive made from silicone gel that will last for 30 days, a hypodermic needle made from 316L stainless steel and covered in an antimicrobial coating, and the drug clozapam, which will be loaded at 3 doses.
Our device has diagnostic and therapeutic advantages. The collected data provides information on the glutamate levels that can help develop better treatment solutions in the future. Also, the device provides emergency medication to prevent the onset of a seizure in real time. Because an individual cannot detect a glutamate increase on their own, the sensor would recognize it and deliver a set dosage of clonazepam prior to the onset of a seizure. This improves quality of life by making it something the patient does not have to think about. Also, using 3D printing allows for greater variability in the device design. A main disadvantage is the risk associated with intracranial surgery. Additionally, any medication comes with a set of potential risks and side effects. Side effects of clonazepam include fatigue, ataxia, depression, memory loss, and liver function. Also, due to size limitations, only 3 doses of clonazepam is being added to the pod, so this would need to be replenished by the patient over time. One final limitation to this device is that the immobilized enzyme driving the continuous detection of glutamate concentrations would need to be replenished over time as well.
Efforts will be made to drive down material and manufacturing costs through the use of 3D printing when possible, specifically for PDMS, PE.PSS, and COC. Other methods for a lower cost will be the use of common materials in the device industry, such as 316L stainless steel, HDPE, and UHNWPE. The greatest cost associated with our device will be the surgical implantation of the biosensor, though we plan to mitigate this through a robotic procedure. Additionally, our device and associated surgery would likely be covered by health insurance. Still, the direct cost of our device is expected to be similar to competing nerve stimulation devices. However, when considering that our device has the ability to stop seizure occurrence and can contribute to valuable research on glutamate epilepsy relations, our device still holds an advantage.
Next, we will discuss engineering criteria and anticipated tissue responses. Our device will have 3 primary functional requirements. First, our device will be implanted to detect and read glutamate levels in the brain. The device will take readings continuously in real time through interacting with the biofluids. The data will be analyzed to determine the glutamate threshold, which will be used as a baseline level for therapeutic release. Next, the electronic receiver system located on the drug delivery system should be able to detect the concentration of glutamate when it has reached maximum threshold. Finally, the drug delivery system will mechanically trigger the release of clonazepam at the threshold. Moving on to tissue interactions, the subarachnoid space is mainly comprised of connective tissue containing cerebrospinal fluid. The immobilized glutamate oxidase layer of the working electrode will catalyze the reaction in the first equation when glutamate is present in the biofluid in the brain. The platinum polymer layer of the working electrode would then utilize the hydrogen peroxide produced from the enzymatic catalysis reaction. The platinum nanoparticles would oxidize the hydrogen peroxide through a reaction in the second set of equations. The result of this reaction creates an anodic current where electrons are entering the electrode. The number of excess electrons entering the electrode is proportional to the number of glutamate molecules in the biofluid. In addition, we anticipate that, similar to other biosensors, our sensor will experience biofouling, or the accumulation of protein cells and other biological materials on the surface of the device over time. Biofouling reduces the sensor's biofunctionality as it encapsulates it during the wound healing process. To prevent encapsulation in our design, we will use a coated bilayer of collagen hyaluronic acid to improve biomimicry and biocompatibility and reduce the foreign body response.
Next, we will outline the assays and experiments to be conducted in the testing process of our device.
There are numerous mechanical and functional tests that need to be run to ensure that our device is safe to be used in patients. For this device, a wide range of functional testing will be done to validate the appropriate release of clonazepam. It is also important to run impact testing on the drug delivery device. This is done to ensure that the device is not compromised during a seizure where it may be hit. Additionally, mechanical testing should be done on the biosensor to ensure that it can withstand manipulation by the surgeon during implantation. The ASTM and ISO standards that are outlined in the table will be followed to test our device. They will allow us to test for injection time, leakage, non-coring, needle shield removal force, needle penetration force, extreme optimum conditions, and extractables and leachables.
For our glutamate sensor to be approved by the FDA, it must undergo rigorous testing to meet the ISO 10993 standards for biomedical evaluation in medical devices. As our device is designed to be permanently implanted and in contact with blood, although indirectly via the cerebrospinal fluid, these tests run the gamut, as you can see from this table. Of particular interest is our device's biocompatibility in the highly collagenous environment of the subarachnoid space. We want to ensure minimal interference with the normal trabecular architecture and the flow of CSF. This would be achieved using neutral red assays and fibroblast cell cultures to determine the cytotoxicity of our materials. It would also be imperative that our materials are 100% hemocompatible, as any thrombogenic or hemolytic activity near the brain could have dire consequences.
For electrical testing, we will perform a functionality test to ensure that the sensor accurately detects the concentration of glutamate. The test will be performed in vitro, where we inject different concentrations of glutamate into a petri dish with our sensor. Over time, we will add concentrations of glutamate up to each concentration at each time point. The sensor will transmit the signals to the electronic display, which will show the concentration of glutamate over time. Therefore, we will determine whether the displayed concentrations align with the known inputs. We will perform functionality tests at increasing and decreasing concentration rates and perform multiple trials to statistically analyze the functionality of our sensor. Next, we will verify that the device only responds to glutamate and there is no interference by other neurochemicals. To do this, a concentration of the neurochemicals shown on this slide will be injected into the electrochemical cell in vitro over time. We will measure the current produced from the oxidation reaction by the working electrode. As shown in the graph, we expect that the curve will be flat for all the neurochemicals other than the glutamate, where the slope should increase.
In the past, researchers have used rodent models to study therapeutics for drug-resistant seizures. We will be using a rodent model to implant our glutamate sensor into the brain. Once implanted, the rodents will be able to roam freely, recording glutamate levels and GABA concentrations in the brain over time. 3 groups will be run with the time that the sensor has been implanted, increasing for each trial. The rats will be examined after 1, 4, and 8 weeks. The rodent model is often limited due to the difficulty in creating small-scale devices for implantation. We will mitigate this complication by using 3D printing, which allows us to appropriately scale our implant. After observation in the brain, we will be using ASTM standards to assess the effects of the conductive polymer on animal tissue, as well as evaluating leachables from the conductive polymer implant. Also, it's important to note that while rodent models are often used, a more complex mammalian brain offers significant advantages. For this, we will be using a porcine animal model. Pigs are cost-effective, have a convoluted cortex, and present an anatomy similar to the human brain. With the large-scale animal model, we will be able to study our sensor as well as the drug release device. A similar surgical procedure will be conducted to implant the sensor in the pig's brain, and at this time, the drug delivery component will be placed on the animal's back.
Device components will be sterilized differently dependent on their material and risk of contamination. The biosensor will be sterilized by ethylene oxide using values found in the study sterilizing a similar coated biosensor for reading glucose. Drug delivery base and pod will be sterilized using a gamma radiation at dosages safe for HDPE and COC respectively. Lastly, the receiver component of the drug delivery system will not need individualized sterilization apart from normal safety procedures, as this component is purely electronic and does not come into contact with the patient physically.
Thank you, and we look forward to your questions.